Hey everybody, in this video we are going to talk about a little bit about experiments with cells from people with a genetic disorder called Xeroderma pigmentosum. So Xeroderma pigmentosum. Okay, so what is this genetic disorder? So individuals with this genetic disorder are extremely sensitive to UV light. So much so that, they shouldn't, ever be out in the sun. And children with Xeroderma pigmentosum, there are special camps called, one of them I believe is called Camp Sundown, where the children play at night outside rather than during the day. So extremely sensitive to UV light, and sunlight has UV light. That's why we all wear sunscreen when we go out in the sun on the beach in the summer or something like that. So in the 1960s, a doctor or scientist, I don't know if he was more of a doctor or more of a scientist, but Dr. James Cleaver was studying fibroblast cells. So fibroblast cells, so this is a type of somatic cell, and he's studying these cells from XP patients and, let's say, non-XP patients, or maybe there are other patients, or maybe they're just volunteers, healthy volunteers who don't have the disorder that are donating cells. So he's studying fibroblast cells from various XP patients, and we'll call these guys non-XP patients. And what he found was, when you took these cells and grew them in culture, and earlier in the course we saw that you can grow fibroblast cells. They can divide in culture up to 50 times, right? And it had something to do with they couldn't go past 50 because their telomeres were shrinking. But so he can take the cells and he can grow them in culture and expose the cells to UV light. So, and when he did that, the cells from the non-XP patients, the controlled patients, well, they performed this process called unscheduled DNA synthesis. So UDS. So what is unscheduled DNA synthesis? And at the time they didn't really know. And I'll explain what this is actually, what UDS synthesis is in a moment, but just know that it's not DNA synthesis associated with DNA replication in S phase. So the normal, how we have to replicate our DNA every time a cell divides, it's not that type of replication. So it's a different type of DNA synthesis that occurs in cells in response to UV light. So, well, the UDS occurred in the cells from the healthy controls when they were exposed to UV light, but UDS did not occur in the fibroblast cells from the XP patients when they were exposed to UV light. So he found something different between these two. And the idea is, well, maybe he could figure out why people with Xeroderma pigmentosum are so sensitive to UV light, if he can figure out why these cells can't perform this UDS. He doesn't know exactly what UDS is, but if he can figure out what it is, we might be able to figure out why these individuals are so sensitive to UV light. So first thing a geneticist would do when we have a phenomenon like this is to determine the number of genes. So how many genes, before trying to figure out, where the mutations are, we can kind of figure out how many genes are required for the process. So in this case, UDS. How many genes are required? And in order to know how many genes are required, or to figure this out, we need to determine the number of complementation groups. And this is a word that we haven't covered yet, and towards the end of this video, I'm going to explain what this is. Essentially, if we can figure out the number of complementation groups, we can get let's say the minimum number of genes required for the process. It will tell us that if we can discover 3 complementation groups or 4 complementation groups, then we know there are at least 3 or 4 genes required for this process. So, okay, so what is he going to do next? Well, what he did, I haven't read his research directly, and this is indirect from our recommended textbook. So he has 7 XP patients, or cells, from 7 different individuals who have this disorder. And I'm assuming they're not from the same family. So 7 different unrelated individuals who have this disorder. So patient 1 provided the fibroblast cells we're calling XP1, patient 2 provided XP2, and so on and so forth. And what he did was he used a technique called somatic cell hybridization. So somatic cell hybridization. And what this is, and we can also call it fibroblast hybridization. So somatic cell hybridization, what is this? We're taking any somatic cell, 2 somatic cells, and sticking them together. Fibroblasts are a type of somatic cell, so that's why we can all call this fibroblast hybridization. So what do I mean by that, sticking them together? Actually, that phrase is not quite accurate, because you're not getting them to stick to each other, you're fusing them. And fibroblast cells, I'm told, have these pointy membranes here. So I draw my fibroblast cells with points. So let's say this is an XP1 cell here. And am I recording here? Yes. And let's say over here, let's say this is a cell from an XP7 patient. And I'm drawing the pointy membranes, characteristic of a fibroblast cell. Now we already know, or Dr. Cleaver already knows, that when he exposes these cells alone to UV light, no UDS. So UDS does not occur. Now he fuses them, or hybridizes them, and then what we get is something we haven't seen in this class, in this course yet. We get a cell with 2 genetically distinct nuclei. I don't know why I draw my 7s like that. One of my uncles did, and when I was younger, I saw him doing this, and I was like, why are you doing that? And he's like, well, it makes the 7s not look like 1s. And I was like, well, okay. So I've been doing it ever since. So where was I? Okay, so we have 2 genetically distinct nuclei here, and they share a cytoplasm. So the cytoplasm is shared. Cytoplasm, nucleus, nucleus. And we can call this a heterokaryon. So a cell with 2 different nuclei, or at least 2 different nuclei, could have more, could have 3 different nuclei, but in this case, 2 different nuclei is a heterokaryon. So when he formed a heterokaryon by fusing cells from XP1 patients and XP7 patients, what he got was something like this, and when he exposed this to UV light, yes, UDS worked. Okay, so what's going on here? How does that happen? So essentially, the genes in the XP1 nucleus complement let me see, genes in XP1 nucleus, let's say, and XP7 nucleus complement each other with respect to UDS. Okay, so what do we mean there? So again, summarize XP1, genes alone, no UDS. XP7, genes alone, no UDS. So, but when we put them together, we get UDS. So, okay, so here's a hypothesis. So let's assume there are two genes, gene A and gene B. And gene A makes a protein that looks something like this. And gene B, well, holds the information for a protein that looks something like this. And these two proteins in a normal cell come together to make a complex. And they fit together, that's why I drew them like that. And say this complex is required for UDS. Okay, so gene A alone, no UDS. Gene B alone, no UDS. They come together, and this fulfills a role in UDS. So if this is true, well, possible genotypes for the XP1 cell and XP7 cell are as follows. So let's say XP1 may have a functional gene A, but it might be homozygous null for gene B. So it has the information needed to make this protein, but not this one. So those cells don't perform UDS, no UDS. This guy over here, XP7, it may have this genotype. So it does not have the information needed to make this protein because both copies of gene A are let's say, null mutants. But it does have the information needed to make protein B. But it needs both, right? It needs XP7, it needs both of these to do UDS, so no UDS. However, when you put these together, well, XP1 provides this, XP7 provides this, and we get UDS. So briefly summarize that. And I didn't draw my cell membrane pointy here, but I think you just imagine this is a pointy fibroblast cell. Here is the two nuclei, one from XP1, one from XP7, and the genotype from XP1 we are predicting is this, two big A's, two little B's, and this one over here we're predicting two little A's and two big B's. So this nucleus over here has the information needed to make this protein. And remember, in the shared cytoplasm, this is a nice review, in the cytoplasm, that's where proteins are made. That's where the messenger RNAs go to be translated into proteins. So the messenger RNAs are transcribed in here and then exported out. So XP1 provides the information needed to make this protein. XP7 provides the information needed to make this protein. Okay, and they can come together, and now the heterokaryon can perform UDS. So whatever this does, this complex, we can say this complex must be needed to perform UDS. And this is just a model. This is just a model to explain how these two genotypes of XP1 and XP7. You know, my ones do look like 7s, don't they? even with the little hash mark. Genotypes of XP1 and XP7, how they complement each other with respect to UDS. So, okay, what can we do with this? What can we do with that information? Well, if you were to look through the notes, I hope you're reading the notes, but if you were to look at the notes, you will see a nice table, and this table is from, it's similar to the one from our recommended textbook, and it's available in the notes. So, and what you might notice, what we have here is cells, or the individual cell types used in the experiment. So cells from 7 different individuals with Xeroderma pigmentosum. We don't have any cells from healthy individuals in here, so this is only a table of cells from the Xeroderma patients. Now, notice there are no cells, there are no, yeah, I guess these are cells too, so we have cells from your body and cells in a table. It's a little confusing, but these are not, there's no information in these. Why? Because this is a mirror image, essentially, and if I was to fill these out, well, you would be looking at 2 places, and it's totally not necessary. So we only have the information put down here. And what is this information? Essentially, if there's a negative here, there's a minus sign. What that means is UDS is not observed when you make a heterokaryon of the 2 cell types. So if you put XP1 with XP2, fuse those together, make a heterokaryon, shine UV light on the heterokaryon, no UDS. Same thing with XP1 and XP3, no UDS. But when you take XP1 and XP4, yes, UDS does work. So by looking at this table, what we can do is we can come up with 3 different groups. And you can try and I encourage you to pause for a minute and try and organize this data, say, into groups of cells that do or do not complement each other. That's a little confusing. So what I'm going to do is I'm going to show you how I would do that. So by studying the table, I'm able to see there are 3 different groups. Group 1 includes 3 individuals. It doesn't matter whether I call them A, B, C, 1, 2, 3. It doesn't matter the order I put these in, but there is at least one group that has cells from XP1, XP2, and XP3 in it. And what does this mean? So these cells right here, if I make heterokaryons between any of these cells, so between XP1 and XP2, between XP1 and XP3, or between XP2 and XP3, no UDS is observed. So these cells do not complement each other with respect to UDS. So we're going to put them all in the same group. XP3, let's say another group over here, I'll put this group over here, XP5, XP6, and XP7, same thing. None of these cells complement each other with respect to UDS. However, if we take a cell from group 1 here and hybridize it with a cell from group 3, yes, we will observe UDS. So within a group, no complementation. Between groups, yes, we do see complementation. And then we have just one cell type in here, XP4. So XP4 will complement UDS when combined with any individual from group 1 or any individual from group 3. So there's no other cell type of those 7 cells that when you put it with XP4, you don't see UDS. So XP4 is all by itself. So in these groups, these are complementation groups. And I think the word's kind of confusing. But this is what we call, these are what we call complementation groups. And essentially the definition of a complementation group is a group of mutants. Could be individuals, could be cells, but a group of mutants where each mutant in group has a homozygous mutant genotype for same gene. And I'm gonna qualify this. I'm gonna say no. You know, I haven't really thought of complementation groups with respect to gain of function mutations or dominant mutations or anything like that. So I'm assuming null and recessive mutations here. But to be simple, just group of mutants where each mutant in a group has a homozygous mutant genotype for each gene. So what does that mean? Okay, so if we have 3 groups, that means there are at least 3 genes required for UDS. And we can say, okay, well, these individuals here, they are all lacking or homozygous mutants for gene A. We can say these over here must be homozygous mutants for gene, I'll say gene C, because they already used gene B for XP7. And then over here, these are homozygous mutants for gene B. So we have at least 3 genes. So now if we can figure out, you know, where the mutation is, well, in these individuals, we can discover gene A and what it does and why it when it's mutated, it causes loss of UDS. And then we can say, oh, is this the reason why XP patients are sensitive to UV light? Turns out that XP patients or XP individuals with XP lack a DNA repair process. DNA repair process called nucleotide excision repair. And, okay, so how is UDS related to this? So UDS is a step in nucleotide excision repair. So in the next video, we'll talk just a little bit about the mechanism of nucleotide excision repair. And okay, I hope this video helped you understand what complementation groups are and how they can be used to discover the minimum number of genes required for a process in a cell. See you in the next video.